What's up, biology students? It's me again. In our last video, we talked about mitosis and how it fits into the cell cycle. Basically, that video was about asexual reproduction in cells. Today, we're going to talk about a related process called meiosis, the process responsible for forming the cells necessary to conduct sexual reproduction. To understand why meiosis is special, we need to be able to compare what happens during meiosis to what happens during mitosis. Mitosis, you should remember, is the process by which the cell's nucleus divides. During mitosis, duplicated chromosomes in the nucleus are split apart and organized so that each new nucleus has the same set of chromosomes as the original. The process is usually paired up with cytokinesis, which is the process by which the cell's cytoplasm is divided, splitting the cell in two. Each new cell will contain one of each new nuclei formed during mitosis. You should also remember that humans have two of every chromosome. We call these homologous pairs, and we would also say that human cells are diploid as a result. The term ploidy refers to the number of sets of chromosomes in the cell of an organism, and ploidy is often represented by N, where N is the number of sets of chromosomes in the cell. If a cell has only one set of chromosomes, or one of each chromosome, we would call that a haploid cell, which is often abbreviated as 1N or just N. A diploid cell, containing two of every chromosome, is abbreviated 2N. A triploid cell with three of every chromosome is abbreviated as 3N, and a tetraploid cell containing four of every chromosome is abbreviated as 4N. In humans, all of our non-reproductive cells, called somatic cells, are diploid, or 2N, meaning they contain two of every chromosome. Our reproductive cells, called gametes, are haploid, or N. That way, when you put two haploid gametes together and combine their chromosomes, you make a diploid cell with two of every chromosome, which is how most of the cells in our body are configured. Meiosis is the process responsible for making these gametes, and during meiosis, the number of chromosomes in the cell is reduced by half. By contrast, during mitosis, the number of chromosomes before and after division remains the same, forming new cells with the same amount of genetic material as the original cell. The major differences between mitosis and meiosis are depicted here in this figure. Whereas mitosis results in the formation of two diploid cells, meiosis results in the formation of four haploid cells. Let's take a look at how this works. In many ways, meiosis is kind of like mitosis times 2, but there are some subtle and significant differences as well. Here on this slide, we see the first half of the process, called meiosis 1. Our example is going to use a very simple cell, one with only 4 total chromosomes, or 2 homologous pairs of chromosomes. Here, in interphase, we see that our cell has 4 unduplicated chromosomes, 2 long and 2 short. After DNA replication, at the start of prophase 1, our cell has 4 duplicated chromosomes, each with 2 identical sister chromatids. During metaphase 1, homologous chromosomes line up along the center of the cell. Here, we see an interesting thing happening, a thing called crossing over. Crossing over is when homologous chromosomes swap genes. This is called genetic recombination, and we can see here that for each of our homologous pairs, part of the dark green chromosome ends up on the light green chromosome, and part of the light green chromosome ends up on the dark green chromosome. This swapping, or recombination of genes that occurs during meiosis can increase the genetic variation within a population, because it can result in new combinations of alleles existing together on the same chromosome that may not have existed before. This is also part of the reason why children are both similar to their parents and unique in their own way, because of the recombination of genes that occurs during meiosis 1. Crossing over doesn't occur during mitosis, and that's one of the major differences between these two processes. Another difference occurs during anaphase 1. During mitosis, the process we learned about in our last video, sister chromatids are pulled apart during anaphase, so that each new cell gets only one sister chromatid from each duplicated chromosome. During meiosis, sister chromatids remain attached, and homologous chromosomes are separated, so that one duplicated chromosome from each homologous pair goes in one direction, while the other homologous chromosome goes in the opposite direction. During telophase 1, these two sets of chromosomes, each containing half of a homologous pair of chromosomes, are repackaged into 2 new nuclei. Cytokinesis splits the cell into two new cells, and these cells enter prophase 2 and the second half of meiosis. The two new cells enter meiosis 2 without going through a second round of DNA replication, and this is what ultimately results in cells with half as many chromosomes as normal body cells. But let's not get ahead of ourselves. After meiosis 1, we're left with two cells, each with two duplicated chromosomes, but only one chromosome from each of our original homologous pairs. From here, everything works pretty much as it would during mitosis. Duplicated chromosomes line up across the center of the cell during metaphase 2, then spindle fibers attach to each chromosome at the centromere, pulling apart the sister chromatids. During anaphase 2, these separated sister chromatids are pulled to opposite ends of the cell. During telophase 2, these chromosomes are packed into two new nuclei. Each cell divides by cytokinesis, and we are left with 4 cells, each with 2 unduplicated chromosomes. We started with one diploid cell that contained 4 unduplicated chromosomes, and we ended with 4 haploid cells that each contained 2 unduplicated chromosomes. We call these 4 cells gametes. During sexual reproduction, 2 gametes combine to form a new cell called a zygote. Each gamete contains a half set of chromosomes, or 1 chromosome from every homologous pair. In humans, this means that every egg cell and every sperm cell contains 23 individual chromosomes, so that when a sperm cell fertilizes an egg cell, and the chromosomes from each combine, the resulting zygote will have 23 pairs of chromosomes, or 46 individual chromosomes, which is exactly how many chromosomes a human being should have. As a result of sexual reproduction, each of our cells contains chromosomes inherited from both of our biological parents. 1 chromosome in each homologous pair is inherited from our biological mother, and the other chromosome is inherited from our biological father. We are literally a combination of our parents' genes, and their genetic legacy lives on in us as a result. When and if we reproduce, we will pass those chromosomes onto our offspring in the same fashion. And in organisms like us, this is all made possible by meiosis, which cuts the number of chromosomes in half, producing cells that must be combined with other cells in order to form a complete set of genes and a new organism that is unique from its parents. Because of meiosis and sexual reproduction, we are more than just clones of our parents, even if we do carry their genes and share many of their traits. Meiosis in and of itself is not sexual reproduction. But meiosis does make sexual reproduction possible, and in organisms that reproduce sexually, each generation of organisms is made of a combination of genes inherited from the previous generation of organisms. This process has its advantages because it helps to maintain genetic diversity in a population of organisms, meaning that those organisms are more likely to be able to cope with changes in their environment than they would if they were just clones of the previous generation. We'll talk more about that part of the story later in the year, but for now, I'm going to bring our video to a close. Thanks for watching, and remember, you can go back and watch this video as many times as you need until you feel like you understand how meiosis plays out, and how this process contributes to sexual reproduction, and how this process is both similar to and different than mitosis.